ELISAs are a type of immunoassay that are commonly used to quantify levels of a specific target within a sample. Samples routinely used in ELISAs include serum, plasma, cell culture supernates, cell lysates, saliva, tissue lysates, and urine. ELISAs are usually run in 96-well microplates, coated with a capture antibody specific for the analyte of interest. Upon incubation with experimental samples, standards, or controls, the target analyte is captured by this antibody. A conjugated detection antibody that binds to a different epitope on the target analyte is used to complete the sandwich. A substrate solution is subsequently added to produce a signal that is proportional to the amount of analyte bound. ELISAs can have different formats. Types of ELISAs. The four main types of ELISAs are direct, indirect, sandwich, and competitive. Each type of ELISA has its own advantages and disadvantages. In a direct ELISA, an antigen or sample is immobilized directly on the plate and a conjugated detection antibody binds to the target protein. Substrate is then added, producing a signal that is proportional to the amount of analyte in the sample. Since only one antibody is used in a direct ELISA, they are less specific than a sandwich ELISA. When to use: assessing antibody affinity and specificity. Investigating blocking or inhibitory interactions. Advantages: fast and simple protocol. Disadvantages: less specific since you are only using one antibody. Potential for high background if all proteins from a sample are immobilized in the well. An indirect ELISA is similar to a direct ELISA in that an antigen is immobilized on a plate, but it includes an additional amplification detection step. First, an unconjugated primary detection antibody is added and binds to the specific antigen. A conjugated secondary antibody directed against the host species of the primary antibody is then added. Substrate then produces a signal proportional to the amount of antigen bound in the well. When to use: measuring endogenous antibodies. Advantages include amplification using a secondary antibody. However, there are disadvantages, such as the potential for cross-reactivity caused by the secondary antibody. Sandwich ELISAs are the most common type of ELISA. Two specific antibodies are used to sandwich the antigen, commonly referred to as matched antibody pairs. The capture antibody is coated on a microplate, the sample is added, and the protein of interest binds and is immobilized on the plate. A conjugated detection antibody is then added and binds to an additional epitope on the target protein. Substrate is added and produces a signal that is proportional to the amount of analyte present in the sample. Sandwich ELISAs are highly specific since two antibodies are required to bind to the protein of interest. When to use: determining analyte concentration in a biological sample. Advantages include highest specificity and sensitivity, and compatibility with complex sample matrices. However, disadvantages include a longer protocol and the challenge of development. Competitive ELISAs are commonly used for small molecules, when the protein of interest is too small to efficiently sandwich with two antibodies, similar to a sandwich. ELISA, a capture antibody, is coated on a microplate. Instead of using a conjugated detection antibody, a conjugated antigen is used to compete for binding with the antigen present in the sample. The more antigen present in the sample, the less conjugated antigen will bind to the capture antibody. Substrate is added and the signal produced is inversely proportional to the amount of protein present in the sample. When to use: determining concentrations of small molecules and hormones. Advantages include the ability to quantitate small molecules. Disadvantages include less specificity since you are only using one antibody, and the requirement of a conjugated antigen. ELISAs tend to be the most sensitive immunoassays due to the binding characteristics of the antibodies and the amplification or different readout systems used. Sample volumes can also be adjusted when you have a very low abundant protein. As discussed above, indirect ELISAs allow for the amplification of signal by using a secondary antibody. Other amplification systems can also be used in ELISAs to make high-sensitivity ELISA kits, where an additional amplification step is used to increase the sensitivity. Examples of these extra amplification systems are alkaline phosphatase or streptavidin HPR polymer.